How the uterus senses labor. New insights into childbirth mechanics. New research from Scripps Research, published in Science, reveals the molecular mechanisms behind how the uterus detects and responds to physical forces during childbirth. This discovery not only explains why labor sometimes slows or starts prematurely, but also lays the groundwork for improving treatments for pregnancy and delivery complications. The body's pressure sensors. The uterus expands dramatically during fetal growth, reaching peak pressure during delivery. Scientists have now identified specialized sensors that interpret these forces and coordinate muscle activity. Artem Patapoutian, whose previous work won him a share of the 2021 Nobel Prize in Physiology or Medicine, led the study. His team found that the body relies on pressure sensors to translate physical cues into coordinated contractions. These sensors are ion channels built from proteins called piezo-1 and piezo-2, enabling cells to respond to mechanical force. The researchers discovered that these proteins play distinct but complementary roles in labor. Two sensors, one process piezo-1 operates within the uterine muscle itself, detecting rising pressure as contractions intensify. Conversely, piezo-2 is located in sensory nerves of the cervix and vagina. It activates as the baby stretches these tissues, triggering a neural reflex that strengthens uterine contractions. Together, these sensors convert stretch and pressure into electrical and chemical signals, synchronizing contractions. The study shows that if one pathway is disrupted, the other can partially compensate, ensuring labor continues. What happens when sensors fail? Experiments using mouse models confirmed the importance of these sensors. Mice lacking both piezo proteins exhibited weaker uterine pressure and delayed births. This shows that muscle-based sensing and nerve-based sensing work in tandem. When both systems were disabled, labor was severely impaired. Further investigation revealed that piezo activity helps regulate connexin 43, a protein forming microscopic channels between smooth muscle cells. These channels enable coordinated contractions rather than independent spasms. Reduced piezo signaling led to lower connexin 43 levels and weaker contractions. Human tissue confirms findings. Analysis of human uterine tissue showed similar piezo-1 and piezo-2 patterns to those observed in mice, suggesting a comparable system operates in humans. This may explain labor problems marked by weak or irregular contractions that prolong delivery. Clinical observations align with these findings. Fully blocking sensory nerves via epidurals can lengthen labor. This confirms that nerve feedback plays a role in promoting contractions. Future implications for labor care. This research opens the door to more targeted approaches to managing labor and pain. If scientists can safely adjust piezo activity, it may be possible to either slow or strengthen contractions when needed. For those at risk of preterm labor, a piezo-1 blocker could supplement current muscle-relaxing medications. Conversely, activating piezo channels might restore stalled labor. The findings also highlight the interplay between mechanical sensing and hormonal control. Progesterone suppresses connexin 43 expression, preventing contractions from starting too soon. As progesterone levels fall near the end of pregnancy, piezo-driven signals may initiate labor. Future studies will map sensory nerve networks involved in childbirth, differentiating nerves that promote contractions from those that transmit pain could lead to more precise pain relief methods without slowing labor. Childbirth is a process where coordination and timing are everything, says Patapoutian. We're now starting to understand how the uterus acts as both a muscle and a metronome to ensure that labor follows the body's own rhythm. This research underscores that the body's ability to sense physical force is essential not only for touch and balance, but also for one of biology's most critical processes.